let's get to the right slides here. I am really excited today because, are you excited too now? All right. Okay, I'm really excited because we can finally, this lecture is going to be centered around talking about epigenetics, which is pretty much my favorite topic in the world. And I also like the topics of dogs and babies as well. But that's not as much science related. Okay, so we left off last week talking about topoisomerases. There's type 1 and type 2 topoisomerases, and they're going to function to hydrolyze phosphodiester bonds of the backbone of DNA. And the difference between these two is that type 1 will hydrolyze the bond in one strand. Is this working? In one strand, whereas type 2, it hydrolyzes the phosphodiester bond in both strands, and then it'll relink those one or two strands together. For the purposes of this exam, you do not need to know this mechanism of how the phosphodiester bond is broken. So you just need to know sort of, what type 1 and type 2 topoisomerase is 2 and the differences between the two. All right. So now we're going to go into epigenetics. And first, let's talk about this problem that the cell has in the nucleus of eukaryotes. So there's a problem in which in a eukaryotic cell, you need to fit about 2 meters of DNA into a nucleus that's about 10 to the minus 5 meters in diameter. And to put it into perspective, that's a lot like trying to stuff 10,000 miles of spaghetti into a basketball. So, right, it's a very, very difficult structural challenges that the cell has, but yet it's able to do it. And the way that our cells are able to solve this problem is to organize our DNA into this into this structure called chromatin. So our DNA will be wrapped around these proteins called histones. These form what's called a nucleosome, and then these nucleosomes can further condense and condense and condense until it forms either the meiotic or mitotic chromosome, which is sort of the most compact form of chromatin. Okay, and so again, this sort of repeats what I just described, is that the eukaryotic chromosome allows the cell to be able to compact a DNA molecule that's about 10 to the 5th microns long into a nucleus that's 5 or 10 to the microns in diameter. And this compaction of chromatin involves these different layers of highly organized folding. So we have DNA that's bound to these little beads of protein that are regulatory spaced, or that's regularly spaced. And so this is what it looks like, sort of at the fundamental, or this is what it looks like at the very fundamental level, where you have sort of this beads on a string organization in which the DNA is bound to these beads of protein on a string. And so these little beads in the DNA that connects to the next bead, that represents what we call a nucleosome. And so the nucleosome is this fundamental unit of organization which allows chromatin to fold into these higher order structures. And so you can imagine that these nucleosomes, they keep compacting until we form this very, very highly condensed chromatin structure, which is the meiotic or mitotic chromosome. Yes, Pat?
Is that how the nucleosomes look when they are in these condensed mitotic chromosome structures?
No, so actually this represents what the nucleosome looks like if you were to take the chromatin in a chromosome and just essentially like pulled it, decompacted it, so just sort of like unwounded it and spread it open. Yes, great question. Okay, so again, nucleosomes are essentially the fundamental units of chromatin. Chromatin is in another way of saying this, or another way of saying this is chromatin is essentially repeating units of nucleosome. So this is the structure of the nucleosome. The nucleosome, by the way, is my favorite crystal structure in the world. I could just spend hours looking at this structure. So this is the structure of the nucleosome. It was actually solved in 1997 by a researcher named Carolyn Luger. And what the structure shows is that the nucleosome contains these proteins called histones. So there's 8 histone molecules, 2 copies of histone isoforms called H2A, so that's yellow, H2B red, H4, which is green, and H3, which is blue. And so you have these 8 histone molecules that sort of form this little compact ball. And then 146 base pairs of DNA wraps around the core of this 8 histone ball. And that's about 1.5 times of 1.5 times that the DNA wraps around the histone. The DNA actually fits around the nucleosome when it's mildly supercoiled. And then the linker DNA, that's about 50 base pairs of DNA. It's not actually shown in the structure, but the nucleosome includes linker DNA, so that's about 50 base pairs of DNA that essentially connects between each octamer of histones that's wrapped by DNA. So for example, if you have, here's double-stranded DNA, and here's some histone proteins, and then this would represent a nucleosome. This would be sort of the next nucleosome. This would be considered a linker DNA. And so actually, the double-stranded DNA with the histones plus this linker DNA, that would be a nucleosome. And then included in the nucleosome is another type of histone called H1. So H1 binds to the linker DNA. Yes, Pat.
Actually, let me just go first.
Sure.
Did you have a question? Oh.
Yeah.
So is the histone a part of the nucleosome, or is
Yes. Yep. The histone. So the DNA with the histones, including the 8 in the octamer, the 146 base pairs of DNA here, 50 base pairs of linker DNA, and H1, that is a nucleosome. Yes, Pat. Okay, that was that the question, or no?
Is the H1 visible in the previous slide where we had that image
Uh-huh.
of the nucleosomes in the little bit extended?
Mm-hmm.
Is the H1 visible in that?
It is not. I don't believe you can see it.
Just because it's too small, or is it just?
I believe that it's because so this is when they try to get the image of the structure of sort of the nucleosome, so these are really sort of repeating units of nucleosomes that are wrapped in DNA. I believe that they actually did not include H1 in the mixture when they try to put the experiment together. But I actually have to double check on that. I mean, I'm pretty sure. I'm 90% sure it's not there. So, great question. Okay. These are great questions. Keep asking questions about epigenetics. I love it. All right. So histones are small, basic proteins. And when I mean basic, I don't mean like, simple. I really mean, they're basic because they have lots of basic residues, right? So histones are found in the chromatin of all eukaryotic cells. E. coli and bacteria, they don't have histones, but they do have histone-like proteins. Their molecular weights are pretty small, so they range between 11,000 to 21,000 Daltons. And like I said, they're very basic. So they're rich in basic amino acids such as arginine and lysine, particularly at the N-terminal region. Now, what's really interesting is sort of within the nucleosome, the N-terminus of each histone, it actually protrudes outwards from the histone core. So this nucleosome actually kind of shows H3, the N-terminus of H3 sort of extruding out, sort of actually in between the two strands of the DNA from sort of the core of the histones. And so the sort of N-terminal regions, these are called histone tails. And they're called tails because they're sort of floppy and flexible. They sort of fold around in solution, kind of like how a dog would wag its tail around. And so actually, because they're flexible, you can't really see them in the crystal structure. So the structure is actually kind of a misrepresentation because the tail is sort of drawn in. It's not actually crystallized. Frank?
Is it just for H3 or do all the subunits have tails protruding?
Great question. It is actually all the subunits have tails protruding. So remember that, right, we have, there's 4 different types of histones, 2 of each, so there's actually 8 histones. The tails from each histone protrude out from the nucleosome. And so this is kind of a model of what it looks like in sort of that, in the nucleosomes with those tails sticking out. Any other questions? Alec?
Just about the H1 histone, what was the significance of that one? Is the DNA also being wrapped around it, or is it just...
It interacts with the linker DNA. It doesn't necessarily wrap it.
Okay.
Yeah. Now, what do you guys think is the importance of why we would have these basic residues on the histone tails? What function would that serve? Michael?
Yeah. Does it supply the positive charge that's needed because DNA is negatively charged and it allows it to wrap more efficiently?
Yes, exactly. Yes. So Michael, is it Mike or Michael?
Either one.
Okay. So Michael really hit the nail on the head, right? So remember that DNA is very, very negatively charged. So negatively charged that, right, DNA actually kind of repulses itself because it's negatively charged along the whole DNA backbone. And so it would actually be very difficult to condense the structure of DNA in the cell without something essentially neutralizing that negative charge. And so that's what those positive charges, so the lysines and the arginines actually does is it sort of neutralizes that negative charge. It interacts, the positive charge of the lysines and the arginines, it interacts with the DNA and then sort of shields the electrostatic repulsion of the negative charge of the DNA. So it allows the DNA to be able to kind of compact even closer due to the ionic charges between the DNA and the histone tail. Pat?
So do the tails then associate with the DNA?
Mm-hmm.
Some kind of
Yeah, they can. Yep, it does actually. So it sort of floats around, and they're not static structures. So it can associate with the DNA, or it can float around in solution and associate with other types of proteins that interact with the DNA and the histones. Okay. All right. So we are going to actually skip this slide and move on to the next slide, which is these 30 nanometer fibers. So the nucleosomes, they can pack into this higher order structure called the 30 nanometer fiber. It's called 30 nanometer fiber because it's literally 30 nanometers wide in the cross section. Now, when you wrap DNA around the nucleosome core, that helps to compact DNA about 7-fold, which is pretty good. It's an order of magnitude of compaction. However, you have to remember that the overall compaction of DNA in a chromosome is 10,000-fold. And so there needs to be even higher order compaction that needs to occur to get it to this 10,000-fold level of compaction above that of just DNA by itself. And so one of the ways that assists that higher order compaction is by taking these nucleosomes and then just really squeezing them together into this 30 nanometer fiber. And that 30 nanometer fiber gives a 100-fold compaction of the DNA. So we're going to skip this slide and talk about the looping of chromatin. So these structures called loops, they are sort of the next level of organization of chromatin beyond the 30 nanometer fiber. And essentially what it is it's taking these 30 nanometer structures and then packaging them into sort of these loop type structures. And very interestingly enough, these loops often contain genes that have related functions. And the loops themselves will associate with this structure called the nuclear scaffold, which is essentially a scaffold that is an assembly of a bunch of different proteins, including a histone H1, as well as topoisomerases. Okay, and so from the sort of the loop structure, the DNA can further even compact even more. So you can have, so really we kind of started with DNA. So that's about 2 nanometers wide. That can compact into nucleosomes. The nucleosomes can compact into 30 nanometer fiber. The 30 nanometer fiber can associate with sort of this nuclear scaffolding, which then can fold even higher order structures that loop onto itself. And then these loops are going to keep coiling and coiling and coiling until we form sort of the most condensed structure, which is going to be the mitotic chromosome. So DNA compaction and eukaryotic chromosomes is going to take the form of coils upon coils upon coils, which then gives it that, what was it, the 10,000-fold compacting over just DNA. And each layer of organization dramatically enhances that degree of compaction. Okay, so that is how we can fit 10,000 miles of spaghetti into a basketball, in relative terms. Okay, so let's talk about heterochromatin and euchromatin. The amount of this higher order compaction of DNA is actually quite variable, and it's the chromatin, or the higher order structures are kind of divided into sort of these two basic classes called heterochromatin and euchromatin. So heterochromatin is chromatin that is really, really highly compact. And so if you were to stain like a chromosome for heterochromatin, you would see that it's very, or for DNA, you would see that it's very highly compact and dark staining. And because it's very, very compact, it's very difficult for the machinery that's involved in transcription to actually access the DNA and to turn on gene expression. And so heterochromatin tends to be transcriptionally inactive DNA, meaning that very little transcripts is going to actually be made from heterochromatic DNA. Euchromatin is not compacted, it's lighter staining, and it's transcriptionally active. So because it's less compacted, it's much easier for the transcriptional machinery to be able to access the DNA and turn the DNA on and perform transcription off of it. So during mitosis, all chromosomes will become heterochromatic, so they'll be highly compacted and ready to be divided. And then during interphase, so that's essentially the part of the cell cycle that is not mitosis, most of the DNA will actually become euchromatin, so they'll decompact, but some of the DNA will remain heterochromatic. And some of these include the centromeric and telomeric regions of chromosomes, as well as the Y chromosome in males and one copy of the X chromosome in females. Okay, so let's talk about you. There is more to you than just your DNA sequence. So there's this kind of a cool article in Time magazine, I don't know, 5 or 10 years ago, that talked about this new sort of frontier in studying gene expression called epigenetics. And it's interesting because what we now know about epigenetics is that your DNA isn't your destiny. There's more to you than just your DNA. Now think about this. The Human Genome Project started in 1990 with the goal of sequencing all the DNA in a human cell. And in 2004, the genome was fully sequenced. So at that point, we knew all of the DNA in your cell. However, the problem, now people wanted to do this for several reasons. They thought if we knew the sequence of our DNA, then we would maybe be able to predict, right, who would develop diseases, okay? What is the underlying basis of someone developing cancer or diabetes or Alzheimer's? Well, simply knowing the sequence of DNA does not inform us which specific genes will be expressed in a cell. And since the expression of a specific gene is often correlated with a disease state, then, right, it's quite difficult to predict a disease based on DNA sequence alone. I mean, there are some diseases that are based on, that are completely genetic, so there's like some mutation that causes disease, but most diseases are not solely they're not solely attributed to a change in the DNA sequence, and it's usually due to the expression of or combination of a number of factors, including which genes are expressed in that specific cell that might cause a disease state to arise. Okay, so every single cell in our body contains the same genetic information but express different genes. So your red blood cells, your bone tissue, your fat cells, they all contain the same genetic content, but it turns out it's these epigenetic events that actually contribute to the pathway by which some stem cell can be converted into some differentiated cell, such as like a fat cell or a muscle cell. So we're going to talk about how how epigenetics actually regulates chromatin and gene expression. Okay, so let's talk about epigenetic regulation and chromatin structure. Epigenetic means to control genes without DNA sequence. And literally it means a layer of regulation over, so that's where the epi comes from, genes, genetics. And so epigenetics includes, first of all, the chemical modification of DNA. So DNA, and there's lots of modification, but for example, cytosine of DNA can become methylated to make methylcytosine. And usually DNA methylation is actually a mark for heterochromatin formation and gene silencing. Yes, Pat?
So why is methylation, I would think that methylation would make like steric hindrance an issue and make it harder to
So that's a great question, and that's because chemically methylation itself does not intrinsically change the structure of chromatin. What's really happens is that there are proteins out there that actually specifically bind the methylated cytosine, and it's the proteins that bind them that actually cause the chromatin to form this more compact heterochromatic structure. So great question.
So it's like a convenient tag.
Yes, it's a tag. It acts as a tag that says, okay, this methyl group is here, and now it's going to recruit these proteins to then condense the chromatin. That's how I actually think about it, is their chemical tags. I just want to make sure I stop at the right time. I want to make sure I stop at 1:10. So that way. Just so that way we save time for the problem solving. Brandon.
Is that very similar to the cytosine to uracil? Cytosine to methyl cytosine, isn't that very similar when cytosine is exposed to uracil?
When cytosine is converted to uracil. No.
It's a ketone, right?
No. Yes. Yes, I believe so. It'd be more like it's probably a little bit more similar to like converting uracil to thymine, because you're adding a little methyl group. Okay, and then the other type of epigenetic modification are histone modification. So each histone has variant forms, and there are certain amino acid side chains that can enzymatically be modified. So you can have methylation, ADP ribosylation, phosphorylation, glycosylation, and acetylation. And these modifications on the amino acid residues of the histone proteins, they affect often the net charge of the residue, the shape of that amino acid residue, as well as other properties of the histones. The modifications themselves are actually going to affect the structural and functional properties of chromatin. So a modification might cause a chromatin to sort of compact even more or to decompact, depending on how it actually affects the charge and the shape of the histone. And because it's actually really affecting the intrinsic structure of chromatin, it also is going to be involved in regulating transcription. So, so here are just some examples of histones that can become modified. Now, typically, it's the residues that are located on these N-terminal tails that actually become covalently modified, also known as post-translational modifications or epigenetic modifications. So, for example, lysines on histone proteins, they can become methylated, so you can add one methyl group, 2 or 3 methyl groups. You can imagine that would increase the hydrophobicity of the lysine, or it can acquire an acetyl group on it, so it can become acetylated. So acetylation actually neutralizes the positive charge of the lysine residues, which influences chromatin compaction. Arginines, they can become monomethylated or dimethylated in the flavors of asymmetric and symmetric methylation. And then residues that have hydroxyl groups, such as serines, threonines, and tyrosines, they can acquire phosphoryl groups. So you can imagine that's actually a pretty dramatic change in the chemistry of the amino acid, because you're taking a polar uncharged amino acid and then throwing a phosphate group that gives it a negative charge, which makes it even more polar. Okay, so if you consider the fact that there are 5 types, there's actually more than 5 types of if you consider all the different isoforms of histones, so there's actually quite a few different isoforms. But H3, here's just H3, H1, H2A, H2B, and H4. and all the different modifications that can occur on the histones, including the tail and the rest of the core structure. There's actually 400 different types of histone modifications. And these modifications can actually occur in different combinations. So you can have an one residue in the tail become modified and another one become modified, and that can actually be a different kind of combinatorial output. So it could be a different functional output in the cell. Okay, so I'm going to skip this slide. There are other epigenetic modifications out there. Okay, and so so the epigenetic or histone code is this code or this wealth of information that's written in our histones. So it's kind of similar to a genetic code or a computer code, where the pattern of histone modifications is going to actually lead to a specific cellular effect or specific downstream event to happen in the cell. So we call this pattern of histone modifications the histone code or the epigenetic code. And again, DNA methylation is involved in the epigenetic code. And so it's thought that you have these modifications, the sort of coded information on the histones, which then is going to act as kind of a language or a signal for controlling gene expression. So, so again, you have a pattern of histone modifications that lead to a particular effect. We call it the histone code. And so you could have, for example, histone H3 that's completely unmodified. It's correlated with silencing. Or you could have certain modifications that take the histones, and that can be, that can then function as being involved in saying, okay, there's methylation at lysine 14 of H3 that's going to turn on transcription, whereas methylation of lysine, well, I can't see it, but if methylation of lysine 9 might be a silencing mark. So here's an example. If you have H3 that's methylated on lysine 4, that's activating, whereas if you have a different lysine on H3, lysine 9, that can be, that's also methylated, that's considered repressive. Let me just look to see how many slides I have, because I have 5 more minutes left. Oh, not very much more. Okay. Where am I? Okay, let me just talk about 2 more things before we go into the example problems. Let's talk about this study, which I think is very interesting, which is this sort of the classic model of epigenetic regulation, and it's these agouti mice. So this was a study that was done at Duke University, now like 10 years ago, which felt like not that long ago, but that was in 2008. And these 2 mice, they're actually genetically identical. So both of these mice, they have the same exact DNA sequence. However, you can see they look quite different. You have a skinny brown mouse, and a fat yellow mouse. And so what's different about these mice is that one represses the agouti gene. So the agouti gene is a gene that actually regulates coat color as well as satiety. So if you express the agouti gene, it actually causes the coat color to turn yellow, and it makes them want to eat more. So this mouse represses the agouti gene, whereas this mouse expresses the agouti gene. So it causes this mouse to have a yellow color and eat more and become fat. And the reason why these mice are identical and yet look so different is because they just have a difference in sort of their epigenetics. So this mouse actually has methylated cytosine in the agouti gene, whereas this yellow fat mouse actually has cytosines that are not methylated in the agouti gene. So you can see that really just the mere absence or presence of the methylation causes the difference in an expression of these genes, which then causes the mouse to have different phenotypes. That is, a change in the coat color and then a change in their body weight. So we're going to skip this slide, and then just talk about how epigenetics can alter chromatin structure. So remember that chromatin is very, very compact. When it's in sort of this compact conformation, it's very, very hard for, like I said, the transcriptional machinery to be able to access the DNA and turn the genes on. So in an epigenetic event, such as the acetylation of lysine, can actually cause the chromatin to decondense and to have more of an open, active conformation, which then the transcriptional machinery will be able to better access the DNA and then turn that gene back on. And there's actually, it's actually, not random. So, the histone acetylation itself is actually working in sort of a by sort of disrupting that ionic interaction between the lysine and the DNA, right? So it's really affecting the charge interaction between the DNA and the histones that causes the chromatin to go from being condensed to becoming decondensed and forming this more open chromatin structure. Okay, so we're not going to talk about this, but this is sort of related to, Pat, what you're talking about, which is that a modification can be sort of recognized by another protein, and then that causes a change in basically gene expression. But this will not be tested on the exam. Okay, and then the rest of the material about how bacteria DNA is highly organized, we're going to go ahead and skip that information. But you guys are welcome to read and learn about this information on your own. Go ahead.
Dr. Alba,
Yeah.
will we turn this chapter 24 notes